All right. Hey, everyone. Cody Locke. I'm Chief Innovation Officer here at the Center for Advancing Innovation. Today, we've got the great pleasure of learning from Dr. Gregory Charlop. He is a man of many talents. He is a trained anesthesiologist. He did his pediatric anesthesiology fellowship at Stanford, at USC. His anesthesiology residency at Stanford and med school at UCLA. He's the author of Why Doctors Skip Breakfast. He's a regular on local TV news, ABC, Fox, CBS. If you watch LA News, Sacramento News, Austin News, you've seen him. He's also founder of Retired Athlete Health. He's founder and host of the Real Estate Flash, top daily real estate show on Amazon Alexa. Has been a physician at Kaiser Permanente, a great group based out of Oakland. So many talents, so much to learn from you, Greg. I'm going to pass it over to you. Really appreciate your time today.
Thanks for having me here, Cody. It's great to be here. And I'm just going to get this out of the way because I know this is not actually a Zoom background that you're seeing. I'm actually speaking to you from my car, and I'm in a McDonald's parking lot, sort of in between kind of California and Arizona. And this happened because I had a COVID exposure in the
Oh, did we lose you, Greg?
from the car. And I think it's going to be fun. And you'll notice it'll probably be a little less formal maybe than some of your other talks. So bear with me, and hopefully we'll have a good time together on this. So we're talking today about the art of science learning, and it's sort of an open-ended topic. And I like to sort of convey information with stories. And I know that from what I understand, share information in the way that I think works out pretty well. And I find that people communicate well with stories. So I would tell you, so I lived in California for a long time, and anyone who knows California knows that it never rains in California. It's dry as dry can be. And we just moved to Texas, and I've got young kids. Now, my kids are 8 years old. Now, in Texas, it actually rains pretty often, and when it rains, like thunder, lightning, you name it. It's just unbelievable. And so we live in this 2-story house, and I'm on the bottom floor, and my kids are on the upstairs floor. Now, my kids have never experienced lightning, thunder, rain. It's all new to them. They maybe saw it in a book or something, but they've never experienced it. So now here we are in our new house. My kids are farther away from us. We're in a strange place, and then boom, there's like lightning and thunder and stuff in the middle of the night. So my kids are freaking out. And so they tell some relatives, and I'm going to keep their names secret just for confidentiality, but they tell relatives about this. And my relatives say, you know what? Lightning is very dangerous. The lightning could kill you. The lightning, even if you're indoors, the lightning could just shoot right through the roof into the house, and it could zap you, and you should not be upstairs. It's too dangerous. So I hear this and I'm upset, right? Because I don't want my kids coming into my room at 2 a.m, waking me up because of a thunderstorm. I want to sleep. You know, I like sleeping, and I think they should sleep. And I wasn't too happy that my relatives are telling them that they're in danger upstairs during these lightning storms. So I speak to them and I say, listen, I speak to my relatives. I say, listen, we can't be talking to them about the risk of lightning upstairs. It's very, very unlikely to happen. And so they message me back, and they find this article of someone who in 2017 was killed inside a house from a lightning strike. And they're saying that because of this, they should not be upstairs from the lightning. And I said, this is one anecdotal case. These are not scientists. I say this is one anecdotal case of someone getting struck by lightning inside a house. It just doesn't happen. The reason it was in the news is because it's so rare. And so I look it up, and it turns out that each year, somewhere around 24, 25 something Americans get killed in total, indoors and outdoors, from lightning strikes. That's the sort of death toll from lightning strikes. Very, very rare. On the cause of death list, it's really about as close to the bottom as can be. And I tell my relatives this, and they're like, well, not only that, but they're in these metal beds, these metal bunk beds, and lightning likes metal, and the lightning is going to straight through the roof. It's going to hit them upstairs in the metal bed, at least get them out of the metal beds. And I just couldn't believe it. And I said, the risk is so low. And they say, well, if there's any chance of reducing the risk, you should do it. And that's kind of the impasse we had. So my take home from this is people, first of all, I'm going to have sort of three main points of this talk. But my first main point is that the general public really doesn't know too much when it comes to looking at data and making sound decisions based on data. And I say this out of love. These are my family members I'm talking about here, right? They love the kids. They love me. They're great people, but they're not able to weigh the risk of lightning just cruising right through my roof, all right? And they hear one story, and when they hear that, there's nothing I could say. I can't give numbers. I can't do anything, because once their mind is made up that this is dangerous, I cannot convince them otherwise, because the story of lightning going through the roof is so compelling that my numbers and my facts cannot compete with that. It's just impossible. I may get them to be quiet. I may get them to stop talking about it, but they will always worry about this, no matter what I say. I could send them 100 articles and that will not change. And this is a thing about the general public. They glom onto sensational things and they accept them as true. And you cannot reason them out of it most of the time. Let me give you another example, not with relatives, but you hear about shark attacks all the time. Cody, you live on the East Coast. You probably hear about this more than I do, but you hear about these shark attacks. So I would ask you, how many Americans do you think die every year from a shark attack? So I looked this up. I don't know, maybe there's different statistics, but I found where I looked, 1 American dies per year from a shark attack. 1. I mean, what an incredibly small number. But how often do we hear about shark attacks? We hear them all the time. Anytime there's a shark or anything. I saw this big thing on Instagram. I just recently got into Instagram. It was about a shark that attacked a seal, and they made this big thing about how the water turned red. It was like a biblical story. We kill 100 billion sharks a year, something like that. A shark kills 1 American a year, and we make this huge deal about this, all right? And it's the same thing. It's sensational. Sensational things make the news. They capture people's attention, and they change behavior. There's lots of people that will not go in the ocean because they hear of a shark. They won't go swimming. They won't even go by a beach. They won't take their kids there. Their behavior is modified because of this incredibly rare event that's sensational. And again, you could look at the numbers and you could say, well, there's one person dies from a shark attack per year. And you know what they'll say? They'll say, I don't want to be that one person, right? So no matter what information you give, you will not be able to shake people out of this sensational thing. So the first point I want to make is that other people don't know information, and many times you can't use data to talk them out of it. Okay, the second point I want to make is, and now I'm speaking to us sort of more in the scientific community, that we don't know information. All right. So I want to tell you a story. So years ago, I don't know if you guys remember this. I don't know how old you all are, but I remember when reduced fat Oreos came out. And this was a big day in my life. Reduced fat Oreos came out. It was a huge deal. So as a kid growing up, I loved Oreos. I loved them. They were just like the greatest thing. All right. But I was also one of these health freaks. And my parents like telling this story. I was 9 or something at a birthday party. And it's like one of these skating parties. So we're skating around, everybody's having fun. You go to the party room, and in the party room, they had cake. I didn't eat the cake because I thought it was too unhealthy. And this is me as a 9-year-old. So that's the kind of person I was growing up. But I loved Oreos. And then so all of a sudden, reduced fat Oreos come out. Now, at this time, there was this sort of wide held belief that fat was bad. Fat was really, really bad. And saturated fat was terrible, fat was bad, and anything we could do to avoid fat, particularly saturated fat, we should do. And so because of this, I wasn't eating cookies and stuff nearly as much as I should have as a kid. But then these reduced fat Oreos came out. It was just the greatest blessing ever. And so I just binged on these things. I would just have package after package of reduced fat Oreos. I open the thing, I'd have 3 cookies, 10 cookies, and before I know it, the whole package would be gone. And I would do this all the time. And all the while, I had convinced myself that I was being healthy. I was being healthy because these were reduced fat Oreos. What could be healthier than that? So I was having something, I enjoyed it, I felt great about it. It was just what I wanted to do. And it was good for me. And so, I look back at it now and I laugh and I was like, well, what was I thinking? It was totally crazy. And so I want to take a minute and analyze what were the mistakes I made when I got so obsessed with these reduced fat Oreos? What did I do wrong? All right. So the first thing is that I had a pre-existing bias. My pre-existing bias is I love Oreos and I want to eat them. So as someone who loves Oreos and wants to eat them, as soon as I find something that supports my desire to eat Oreos, I am going to latch on to that because that is information I want to have. It supports my bias. It supports my feeling that I want to eat Oreos. And now I could do it guilt-free. And you see this all the time. People that love drinking alcohol, the minute a story comes out that alcohol is healthy, they're all over it. Like take resveratrol and red wine, which, by the way, I think it probably is fine to have a couple glasses of wine. But there was this thing, resveratrol and red wine, and this is super healthy, and this is great, and everybody should be drinking it. To get enough resveratrol to make a difference, you probably have to have like 1,000 glasses of wine. There was sort of a funny story I heard about this. It was like a cartoon. It was a couple. The man was speaking to the woman, and he said, "Oh, honey, did you hear? We should have wine. It has this healthy resveratrol in it." And the woman said, "Well, I think you need to have 1,000 glasses or something to get enough." And then the man says, "The news keeps getting better and better," and because he wants to have 1,000 glasses. So people have, and even people in science, we have these existing biases, and when we find data that supports our view, we glom onto it and we accept it as true. And when we find data that goes against our view, we tend to push it aside or marginalize it or assume that there's something wrong with it, and we discount it. So even scientifically trained people like us are susceptible to many types of cognitive biases, and this is one of my favorites, that we accept data that's true, and I mean that fits our kind of worldview. And the same thing is definitely true in publications. When you look at publications, and this happens all the time, if you're researching something and it's an interesting thing, like let's say you're researching, I know I should have probably thought about this before the talk, but let's say you wanted to find out whether eating at McDonald's reduces your risk of cancer. I'm just making this up. If you did this study and you found it did not reduce the risk of cancer, that study would probably not be published because everybody knows that it's boring. But if you found a study, say, if your study showed that eating McDonald's reduces your risk of cancer, that would be all over everything. It would be huge. And the reason why that would be huge would be because publications, whether they're scientific publications or lay publications, they like publicizing sensational stuff. So if there's 1,000 articles that come out that say that eating McDonald's is not particularly healthy, and then there's one article that says that eating McDonald's is really healthy, that one article will get way more attention than the other 1,000 that says it's unhealthy, because it's attention grabbing, it's headline grabbing. So we as consumers of scientific information have biases, and it affects how we interpret stuff. But even the distributors of scientific information, like these journals, have their own biases in favor of stuff that's interesting, that will sell journals and create press. Now, the second reason why I was wrong with the Oreos is that the data changes. I thought fat was the worst thing in the world, and I didn't consider other things like, well, sugar. And so, Oreos, the reduced fat Oreos, I looked up the nutritional information. They have 50 calories a cookie. 50 calories a cookie. These are little things. You've seen them. They're small. Each one is 50 calories. I probably had, thousands of calories eating this whole package of Oreos. Now, I'm going to get more into data changing in a minute, so I'm going to come back to that. But one reason I was wrong was that the data changed. The third reason I was wrong, which is important, and again, I have a scientific background and I still mess this up, is even in science, we have difficulty weighing risks. Now, if you look at reduced fat Oreos, they're not zero fat. They had 30% less fat than regular Oreos, 30% less. So let's just, for the sake of discussion, let's assume for a second that the science is, the thinking at the time was true. Let's assume that fat really was the bad problem, and that was it. We don't have to worry about anything other than fat. Let's just assume that's true for a minute. These were 30% less fat. So in my mind at the time, I wanted to eat the Oreos, they're 30% less fat, therefore I could eat as many Oreos as I want. But clearly that's flawed thinking, even if you assume that fat is the only unhealthy component in Oreos. It only had 30% less fat, it didn't have zero fat. So I'm thinking, well, there's a little less fat, I could eat as much as I want. I didn't weigh the risk. What I should have thought is, well, if I'm going to eat them, maybe I could eat a little more. That's what I should have thought. Or, well, if I'm going to eat them, maybe I won't feel quite as bad about it. But instead, I took it and ran with it. And I said, oh, 30% less fat, it's no problem. And then I just ignored the fat altogether. I simplified the equation in my mind. And I said, well, 30% fat, well, if 30% less fat, that's like 100% less fat. You know, it's close enough. And so I'm just going to eat these Oreos. So that was another big mistake I made. I was not, as a scientifically minded person, I could not properly weigh the risk of these Oreos. Okay, so that's point two. Point number one, the general public really doesn't know much about science and has trouble looking at data. Point number two, people in science, like myself, often don't know much and have trouble looking at the data and make mistakes. And an example was the Oreo story, and that we have our own biases, data changes, and we have difficulty weighing risks. Now, the third point, and the main point I want to get to for you, is that we need to stay humble. This is a very big point, that we need to stay humble. And it's very easy, especially after point number one, and when we have these conversations with relatives, it's very easy to not be humble and and think, well, you know, my relative doesn't know anything about lightning, therefore I know everything. But we need to stay humble. And I want to give you a few examples of this. So like Cody said, I'm an anesthesiologist, and I work in the operating room, and I keep up with the latest data, and so stuff makes it out in the anesthesia literature and surgical literature. And then, as a cutting-edge anesthesiologist at a big hospital, I like to implement this stuff. So, some years ago, there was this thing that came out called tight glucose control. Tight glucose control. And the idea is that if you have a surgical patient and their blood sugar is high, they're at greater risk of all sorts of complications. They have higher perioperative complications, much higher rate of wound infections, because the glucose sort of impairs the ability of the immune system to act in the site of incision. And it sort of just overall worsens outcomes. So I read this. There was a couple of good studies that came out about it. It made sense because we know that high blood sugar will impair the immune system. And so I'm thinking, wow, this is awesome. I'm really cutting edge. I'm really smart. There are some good studies, and the mechanism for this makes sense to me. So I'm going to start using tight glucose control. And so what tight glucose control means in an operating room is you don't just do the anesthesia, but you have to look very carefully at what someone's blood sugar is, and you have to keep it in a very narrow range. And if it goes up, you treat it with insulin or something like that to bring it back down. The trouble is it goes high, so you treat it with insulin. Now, that sounds great in theory. The problem is that if you give insulin to somebody, especially someone who's not diabetic, their blood sugar can tank, and their blood sugar could really drop. And here's the deal. Low blood sugar is much more dangerous than high blood sugar. Yeah, high blood sugar can increase your risk of wound infections, but low blood sugar will just kill you. And, as an anesthesiologist, my job is not to look at blood sugar every second. I'm doing other things. So yes, I could check blood sugar from time to time. I can give insulin, but it's not the only thing I'm doing. So of course, being me, I took this to its logical extreme, and I had a baby that was having a big surgical procedure. And I'm like, you know what? I'm going to use tight glucose control on this baby, because baby is just going to have an incredible outcome. And so I'm measuring blood sugars, and I'm giving insulin, which, by the way, is not an easy thing to do with infants. And I'm doing all this stuff. And then I'm bragging to the surgeon, how cutting edge I am as an anesthesiologist, because I'm following the latest, all the latest data. So thankfully the baby does fine. But then afterwards, all these studies come out that this type blood sugar control actually is causing people to die left and right. And we're actually having higher mortality now that people are using it. And the reason is what I mentioned, is that, yeah, you're avoiding high blood sugar, which is great, but you're causing a worse problem, which is low blood sugar. And so we're just knocking people off all over the place by making them hypoglycemic. So that's an example where we had some data, there was some research, it makes theoretical sense, but it was bad. There's another example, we had this in anesthesia, not quite as dramatic, but there are these things called beta blockers. Beta blockers are a very common class of drugs. They're used to treat high blood pressure, and they block beta receptors that normally things like epinephrine and norepinephrine bind to. And when you block these beta receptors, it'll slow the heart rate down and it could lower the blood pressure, which is a good thing. And a lot of people, some of you may be on beta blockers, and they're great drugs. But a couple articles came out that were good. They were randomized, like placebo controlled, the whole bit, just the kind of study you want to have. And they showed that perioperative beta blockade, meaning starting people on beta blockers for the purpose of surgery and then continuing it through and after their surgery, was like God's gift to surgery. It reduced strokes, it reduced heart attacks, it reduced mortality. You had much better outcomes. It was just the greatest thing. And some people were saying that we don't even need to do cardiac testing anymore because beta blockers are so great. We just beta block people and we forget about it. And that's it. You do that and your job is done. And so everybody started doing this, and it got into these protocols. And I don't know if any of you work at hospitals, but getting protocols in hospitals is hell. It's just, you have all these committees and you have to do all these things. And then the government was checking to see if people were beta blocking, and it became like a measure of quality. It became this huge thing. Everybody had to do it, and you were scoring on it. And we took a lot of time, and it was a big thing. And now we find out that beta blockers really don't make a difference, except for a very small category of people that were already on beta blockers. You don't want to stop it. But starting beta blockers for surgery probably doesn't make a difference. It probably doesn't help. So a huge amount of time, a huge amount of effort, all these things, people got in trouble for not using beta blockers, and it turned out to be, for most people, worthless. But we were so convinced of its value. And so, this brings up this sort of crisis that we're facing in medicine now, which is this replication crisis. And I'm sure that many of you have heard about it. The replication crisis is that what we're finding is that a lot of stuff that makes its way into the scientific literature, stuff that's making it into journals, even good journals, they're not able to reproduce this. So you'll have a study, it sounds great, it may be just the kind of study you like, randomized, double-blind, placebo-controlled, everything, just the class one, just the exact kind of study you want. But when you do the study again, it doesn't give you the same results. And this is happening a lot. And so there was this survey that was done, and they surveyed something like 1,500 scientists, and something like 70% of them found that they could not reproduce another scientist's work, published work. And something like 50% of them couldn't reproduce their own work, which is pretty breathtaking to me. And so more stuff is coming out now about this replication crisis, and we're finding that this is a big problem. And it's a big problem in several different fields of science. It's a big problem in like the soft sciences, but it's a problem also in like the more hardcore sciences also, is that these things are getting published. They may be great study designs, but they're still garbage, and they're wrong. And who knows why they're wrong? Who knows if the scientists did something wrong? Who knows if it was bad luck? Who knows if they lied and they made something up? Who knows if when they say that they corrected for something statistically, maybe they didn't correct for things properly? Just over and over again, these scientists, either intentionally or unintentionally, were making these big mistakes. They're getting into big journals, and they're not reproducible, and they're probably wrong. But we read these things and we say, oh, well, this is in Nature, this is in Lancet or something, this has to be right, and it isn't. And so it's very important to stay humble. Now, there's a great book. It's not about science per se, but I really recommend it. It's called The Signal and the Noise. It came out a few years ago. It's by Nate Silver. He's sort of one of these famous pollsters. If you're into politics, you'll see him, and he's also into sports. And he's one of these people. He analyzes poll data and stuff a lot. And this problem extends beyond science. It extends to pundits and experts in general. And so, what he did, and he talked about it in the book, and he could explain it better than I can, but you'll find a lot of these experts. You turn on the TV, and I'm not going to get into politics per se, other than to kind of make this point. When you turn on the TV and you see a political pundit, they'll analyze something and they'll tell you what it means. Oh, this happened, so this is what the candidate will do. Or, oh, this happened, so here's what will happen with the polls. This happened, and so this candidate's going to lose or to win. And they will provide their analysis. And they've been around a while. They've been on TV many, many times. They've seen tons of other elections. They've spoken to candidates. They've looked at the polls. They've done all these things. These people are real experts. But they're usually wrong. They're usually wrong. There's no follow-up almost all the time to see whether the predictions that these talking heads on TV make, these experts, to see whether the predictions that these experts make are correct. So what they do is they'll go out there, there'll be some kind of story, they'll opine about this story based on their expertise and their many years in the business and their inside connections. And if they're right, you'll hear them say it next week. Oh, of course I talked about it. I was right. I'm always right. If they're wrong, it just goes down the memory hole. It's forgotten. They just make their next prediction. And this happens all the time. And so Nate Silver in his book, The Signal and the Noise, he talks about this, and they actually did some research on this after the fact, and they looked at people who have made these predictions. Again, this is not in science, but I think it's important to understand this concept of expertise. And these experts were very often wrong. I mean, a huge percentage of the time. And what's interesting is a lot of times the people that were more vociferous, the people that were more clear, the people that were more convinced that they were right, were wrong more often. And he sort of talked about these sort of hedgehogs, I think he called them. They're often sort of partisan people. They have strong initial biases. They glom onto any piece of information that supports their view. They come out there, they make these outlandish predictions. They're totally convinced they're right. They know everybody, they know all the inside stuff, and they're wrong. So it turns out that the lesson I think that we need to get out of this is that we need to be sort of humble, and we need to accept the fact that we may have been around a while. We may have seen many things. We may have talked to people who are on scene. We may have even done research in this area, but we could still be wrong. And so can the experts that we consult with. Another way that we're often wrong is we can confuse clinical significance with statistical significance. And this is a complicated topic. Now, I'm sure that you've all heard about this idea. So, the idea might be that, and you probably all know stats better than I do. So I'm not going to try to compete with you in my statistics knowledge here. But as a medical practitioner, I'd like to share my perspective on this. So you could do a study, and maybe you'll look at a blood pressure medication, and let's say it's just this unbelievable study, and it looks at 100,000 patients, and it's perfectly done, and everything is great, and the results are very uniform, and this new blood pressure medicine, say, lowers your systolic blood pressure by 2 points, so from 140 to 138, and it has a really low p-value, and it's very reproducible, and it's real. And it's statistically significant, p of 0.0001. So this is very statistically significant. But it's not necessarily clinically significant. So lowering someone's blood pressure, systolic blood pressure, 2 points for that patient probably doesn't mean anything. That 2-point decrease in their systolic blood pressure is probably not going to reduce their risk of stroke or heart attack or death or any of the other things that we're trying to prevent when we're trying to lower someone's blood pressure. And as a physician, assuming I've read the data and assuming I know the studies, and I know that without a shadow of a doubt, if I put my patient on this medication, their systolic blood pressure will drop 2 points, then I have to kind of weigh, is that worth the cost of the medication? Because it's new, it's probably expensive. Is it worth the side effects that we know of? Is it worth the side effects that we don't know of? You know, a lot of things come out and they seem great today, but 6 months from now or a year from now, they have some terrible unwanted effect that we don't know. So as a physician, I would be very leery about starting someone on this type of blood pressure medication. On the other hand, though, and this is sort of the interesting wrinkle, so it may be that for my individual patient, starting this medication will probably not help. But it could be that if everybody were on this medication with high blood pressure, it may be that on a population basis, when people's systolic blood pressures overall are reduced 2 points, you might, on a population, reduce strokes by 10,000 a year, reduce heart attacks by 40,000 a year, I don't know. So something may be statistically significant in the lab. It may not be clinically significant for the patient in front of someone, but it may, sorry, it's an alarm in the parking lot here. I thought these things were gone like in the 80s, so it feels retro that they're coming back. I don't know if you can hear it on the microphone. But then it may again be statistically and potentially clinically significant on a population basis. So it's actually, the clinical significance, statistical significance question is an interesting one. And it really depends a lot on what is clinically important and what sort of population size you're looking at. And again, it's a reason to stay humble, because if you have a drug or a product or something that maybe has a very, very small but statistically significant benefit, you may have difficulty selling that to an individual doctor, because assuming they've read your data, they'll know it's not going to make much of a difference for their patient. But you might have success selling that to a larger entity like a government or a health insurance or something, because the overall benefit, when looked at over thousands or millions of people, may actually be beneficial. And obviously it would depend on things like the cost of the product. So I say this to say that depending on how clinically significant your product is, if it has some statistical benefit, it may potentially be worthwhile to look at, but it requires careful consideration. It requires looking at the number of patients you're treating. And it means you have to kind of calibrate who you're talking to.
Hey, Greg, can I ask you a question? Because
Sure.
you may not be able to reading the chat because you are in a car.
Yeah, I can. Yeah.
My question is, are these problems coming up because we always tend to look at one cause, one outcome relationship rather than having a complex interdependence on multiple variables, which is saying, oh, I mean, that's why these fats keep coming. It's low carbohydrate, then it's sugar, and then it's fat, and then it was kale, and then it was pomegranate, and then now it's turmeric. And it's because we just pick up one variable, we look at outcome rather than saying, hey, body is a complex system of hundreds of variables. We need to look at least the top 5 that are significant and see their interplay. So that's point number 1. And number 2 is, anytime these data are published, it was my belief, and I might be completely wrong, that they're peer reviewed, where raw data is provided so that somebody can duplicate and re-verify that the results are indeed true, and then only you get to publish them. When, I mean, it seems like cold fusion from 25 years ago.
Well, I have to tell you, and I'm so happy you asked this question, because I didn't plant you in the audience, but it probably seems like I did. I don't know if you could read this, but the next thing I was going to type, I've got a little old school notebook here because I'm in the car. But the very next topic I was going to talk about was actually that things are complex systems. And so the complex system is my very next topic. So I could almost have planted you as the perfect segue for that. So I will answer that in 1 second. But for your second point, so in these reputable journals, in theory, everything published there is peer reviewed. But peer reviewed doesn't necessarily mean that the study was reproduced. It means that people that are experts in the field have looked at the data, they've looked in theory at the methodology, and they've decided that this study and this methodology makes sense, and that they endorse it. So, we see this all the time, and truthfully, it probably has to be that way for most biological studies, right? Like, let's say there was a study and it took 5 years to do, and it's a prospective randomized study, which is great. That's the best kind of study you want to do. There's no way that someone's going to reproduce that study before that study's published. It just can't be done, because that would mean you'd be sitting on that information for 5 years. So the vast majority of the time, even in peer-reviewed journals, the research isn't repeated. It's just looked at. You don't know how carefully it's looked at. Somebody looks at it, and an expert looks at it, and the expert says, "Oh, this is great," and then it gets published. So there may be some exceptions here or there where things are looked at, but the vast majority of the time it's not reproduced. And this replication crisis that we were talking about earlier, these are in studies that were published, and many of them in peer-reviewed journals. So I think the problem is that we can't take to the bank the fact that because something was peer-reviewed, it's sound or reproducible. And the other thing I should mention, and you probably know this, or if not, you'll encounter this, is it's hard to do a lot of types of studies where you're not testing sort of a proprietary patentable thing, particularly in health. So when you look at things like, say you wanted to test the health benefit of broccoli as an example, it's difficult to do that because there isn't really a big broccoli industry that will fund your broccoli study. But if, by the grace of God, someone funds your broccoli study and you do it, that will never be repeated, because there just won't be enough money to repeat it. You got lucky in having that study in the first place. So a lot of these things on, say, foods and nutritional supplements or lifestyle interventions that can't be patented, those studies usually cannot be, they're hard to do in the first place, and they're even harder to repeat because there just isn't enough money for that. Whereas you might be able to get a couple studies on the hottest new cholesterol drug because there's a lot of money to be made there.
Yeah.
So to get to the complex system, so I love this, and this is a big thing, and I think this is a big problem that we make, which is that complex systems we often discount. Either we forget they're complex, or we don't know how to take the complexity into consideration, and we kind of break things down. We deconstruct them into one point. And the human body, and I think a lot of you are probably in healthcare related products, the human body is really complex, and there's a lot of different compensatory mechanisms. Some we understand, some we don't understand. And you deconstruct things and you say, well, I'm going to make this one change. What will that do? Unless you're able to test it in real humans over time, you don't know how that affects a complex system and what the ultimate outcome will be. And one of my favorite examples of this are artificial sweeteners. Now, there was a time that I was a big fan of artificial sweeteners, another time that I was wrong. And I remember I would drink Diet Coke, and actually everybody at the hospital would. We'd go to these conferences, and everybody would drink Diet Coke. That's all you'd see, Diet Coke. Maybe somebody would have water, but pretty much everybody had Diet Coke. Or I remember then I thought, well, sometimes maybe I want to ease off the caffeine, so I'll have like Diet 7 Up or something. So I just drank gallons and gallons and gallons of the stuff because it has an artificial sweetener. And I'm thinking, well, sugar is bad, obviously. These things don't have sugar. So I could drink as much of this as I want, and it's fine. And they tested it in lots of people. And I know there was some concern at whether they cause cancer, but I thought that risk was pretty low. And so I was willing to take. But now we know that diet sodas and artificial sweeteners are linked to a lot of health effects, including the very thing that you wanted to avoid. They cause obesity. The consumption of diet sodas has now been linked to causing obesity, which is pretty mind-blowing. And it's the opposite of what you would think. And yes, it's true that diet soda has 0 calories. And if I put you on a desert island and the only thing you had were pallets of Diet Coke, you would not get fat. But that isn't the only thing you have, and you're not on a desert island. So you'll drink your diet sodas, but then you're going to do other things, and overall your weight is going to increase. Why does it increase? We don't fully know, but it may be that it changes your intestinal microbiome. Now, 10 years ago, 15 years ago, when we talked about diet sodas, nobody was talking about how artificial sweeteners are going to impact your intestinal microbiome. And even if you did, you'd think, well, who cares? But now we know that your intestinal microbiome is a big deal. It's something that we never really thought of before. And we know that artificial sweeteners affect it. And maybe that makes people fat when they drink diet sodas. Who would have thought of that 10 years ago, 15 years ago? Nobody would have thought of that. It also may do other things like affect your craving. And maybe your brain was expecting something sweet and it's expecting your blood glucose to go up, but your blood glucose doesn't go up and you're disappointed. So now you're on the hunt for a cheesecake. So what we know is that the body is a very complex system, and we've made this mistake over and over again when we deconstruct and we only look at one piece of information, and it has all of these other unintended consequences that we don't fully understand. And the only way you could really know this is if you have a lot of people, you do randomized prospective trials, you follow it over time, which is hard to do for anything except for expensive drugs. And even then you could still be wrong. So my third point is that we need to stay humble, and we may review the data, but we could be wrong because the data may change. We could be wrong because the data could be flawed. We could be wrong because we didn't take our own biases into account. We could be wrong because we trusted experts who really don't know as much as it seems like they do. We could be wrong because we think that something that's clinically significant and statistically significant, it's the same. We could be wrong because we haven't considered how complex the human body is. So there's lots of ways we could be wrong, and we always need to be open for considering the fact that we're wrong about something. And so that's kind of the main points that I wanted to bring up today, and I'd be happy to take any other questions if people have them.
Please, teams, go ahead. Ask some questions. You can take advantage of the chat box if you like, but love to hear you and see your video.
So I have a question about if somebody wanted to start, right? So they are
say that again. Say that again.
Can you hear me now?
I can. Yeah, I just missed a few words. Say it again.
So, say we are doing a startup challenge, and so we need to evaluate the patents. And there are some publication around the patents that inventor have published. So how can we are sure that this product or this particular a chemical is going to work in the, is it a viable product going to be happen? Is it possible to look at the patent or data, or is it we need to make sure that we can't go ahead and we can stop the process of going forward?
Is your question, how could you, sorry, go ahead.
I'm not sure you can understand what I'm trying to say, but
are you asking how do you know whether the product is patentable, or how do you know whether if your product
yeah, there is already patent, but you said the reproducibility is a problem. There's a problem in the process or science, everything.
Right.
We need to create our business around the patent and whatever data right now we have, right?
Oh, right.
How can we be sure? Is there any particular way we can evaluate it, or we have to just wait for some research to be done and then we can find out?
Well, unfortunately, this is one of the problems, and this is a great counterpoint to my presentation here, because what I'm trying to argue through this talk is that a lot of the things we think could be wrong, and it's hard to figure that out. But you bring up a great entrepreneurial counterpoint to this, which is if we were just to wait all day and have tons and tons of research and you keep retesting it over and over again, nothing would ever get invented. And we would miss things that actually are really effective and do work really well. So I'm actually really happy you asked this, because you don't want to fall back in the opposite problem, that you're paralyzed and that you think, well, there's only been 2 studies, maybe there's others that will eventually find this product won't work. And so that paralysis could be very bad, because you may have one of the greatest products out there, and if you keep waiting for more and more studies to be done, you're never going to finish it. You'll never get it to market, and you won't realize its benefits. So I would say to you that I think if there's some preliminary data that something works, I would keep trying to bring it to market concurrently with some testing. And honestly, a lot of these problems you don't find out for drugs, especially until after they've been on the market. And so I would say on balance, I think it's still better to bring it to market or do your best to advance it, because what will ultimately happen is if you take a drug out of obscurity, you take a patented drug, you take it out of obscurity, you do some research, it looks good, then in theory, what should happen is maybe a big pharmaceutical company or something, maybe will pick it up or they'll partner with you or something like that. And they'll have the muscle to do better studies than you ever will be able to do. So I would say if you find something that's promising, I would grab it by the horns, because number one, you don't want to let indecision paralysis stop you from working. But number two, if it's a promising chemical, I'd want to get it in the hands of people that have the money to do research on it as quickly as you can. So I would say you should run with it.
You know,
Thank
and
you.
I would just add, at the Center for Advancing Innovation, we really look for relatively de-risked, federally funded technologies around which to launch startups, and where there has been reproducibility from different labs, you know, different people involved, publication from those groups in different peer-reviewed journals, we give those kinds of technologies preference. Ultimately, we want to increase confidence that, you know, the entrepreneurs can do what they say they can, and, you know, the investors are getting what they're paying for.
Greg, and also there is a process, right? I mean, there are things that are related to life and death, and there are other things. I mean, life and death, you have to make sure, because you don't want one death, period, which is really de-risking. The other thing is, aren't there methods under FDA where you do a staged introduction with a limited folks who, for example, I remember during HIV time, people were dying, right? So those people were ready to take anything to regain their life. That situation is different than, high blood pressure. I mean, it has been there for last 100 years, will continue. So there is no level of urgency of dying. So the method that you choose depends upon the severity and the immediacy of the issue.
I agree. And I think there's several points that come out of what you're saying. I mean, one is obviously, if there's greater potential benefit, you're willing to take greater potential risk. You know, you had said, well, you wouldn't want one person to die. Well, maybe you would be happy with having one person die if it's a drug that will save thousands or tens of thousands of lives. On balance, that one person dying, it's worth the trade. I mean, I'm going to go this so far as to say that every prescription drug has probably killed some people, but you would hope that the benefit of those prescription drugs outweigh those deaths. So obviously you would hope that no one dies from your product, and you'd hope that no one dies from any prescription medication or device, but even the best devices and even the best medications are probably going to contribute to death. So I agree with you in that you need to weigh risks and benefits, and if there's this huge potential upside, you're willing to tolerate more potential risk. I mean, it's interesting, if you look at Viagra, for example, you know, if you'll pardon the pun, we could debate what the upside is of Viagra, right? It's not saving anyone's life, although they do use it sometimes in neonates with pulmonary hypertension. But for the most part, Viagra isn't really saving anyone's life. And some people die from Viagra because they combine it with nitrates and other things and people die. Yet Viagra is still on the market. And, you know, it carries warnings. And in theory, the physician should know this when they prescribe Viagra. So even things like Viagra that aren't necessarily life-saving, I think we as a society are willing to accept a certain risk of death. But obviously your acceptance of the risk of death is going to depend on the potential benefit of the product. You know, in terms of like sort of staged rollouts, yeah, like, you know, there's certainly, you know, there's examples of compassionate use, and you see this a lot either in recent diseases, like for COVID, there may be compassionate use for medications that may or may not help because you have someone in the ICU on a ventilator who's going to die without it, and so we do it. Or compassionate use maybe for orphan diseases, which is a big problem. Orphan diseases, very few people have them. There isn't enough money to research treatments for them. There's a drug that exists maybe for something else that might potentially help, but it hasn't been proven to help the orphan disease. In theory, it might help. And so you might get approval for drugs to treat orphan diseases. And I think, again, you would figure that that's worth the risk because you know there's not going to be any major randomized controlled study to look at these rare diseases.
Hi, Greg. I'm Rene Anand. I'm the CEO of a new company. I was an academician, and I'm the CEO of Neurextam. I've been competing in these races, brain race, and this one too. And I want to change our perspective. I think much of the confusion in the world of medicine is coming from a failure to recognize something very important. The first one of which is we are all different. It is written in our genomic code that we are different. So any kind of placebo-controlled study or anything else is largely bogus, okay? Even something simple like when they put up medical studies, African-American versus Caucasian or white, complete rubbish, because African-Americans derive from a whole bunch of different countries in Africa, and they have some of the greatest genetic diversities. So you're comparing apples to oranges, no wonder the output is rubbish. And I like your cynicism in pointing out why our clinical studies have been failing, because we have been doing p-value studies, and I use the word p cynically with the spell P-E-E, okay? There is no statistics there. The mathematics of statistics demands very strict axioms and assumptions without which they are not valid. I come from the world of physics. I understand statistics like most other mathematicians do, and they've been misused in biology to such terrible degrees, spending billions of monies in all those journals you just talked about, which is why they've never been reproducible. Okay? Now, that's the bad news. That's the last 100 years. The good news is genomic sciences have emerged. They're bringing about an entirely different way to look at how human biology is set up and how do we go from the differences amongst us to making predictive science more reliable. The field of gene regulatory networks has emerged, and you may want to pay attention to that. And it dissociates all the other variables. We've been doing a lot of this stuff, and it requires a lot of math to resolve large data sets, okay? So my company is being founded on really good science first. That is our only criteria, not profits, return on investments, business models. All that is rubbish, because if you build a sandcastle, it's just a matter of time, it'll wash away. And we see that in clinical trials. That costs the companies and the investors billions to begin with. So they get misled. So my thinking is, what I'm trying to say here literally is, there's a new modern medicine that has come about. It is being developed by really good entrepreneurs all across the country and in other places. They are looking at big omics data. They're putting together the foundation of disease. And in our own hands, I study brain disorders, Alzheimer's, autism, drug abuse. I've been funded by an SBIR recently on opioids. Here's the beauty. We found out there are three variables that are coming into play. One is your genes, which cocks the trigger. The second one is the environment. And the third one is nutrition. The interplay of those three gives rise to the clinical threshold that you're going to see for any given individual or any group of person. And until you know those three variables and which of those three variables are playing within that disease space, you won't be able to do any good business in the world of medicine, modern medicine, because the other variables will confound everything you have built your drug on, your predictive science on, or anything else. So we are now coming to the point where, like it or not, it's the way nature is set up. That's the nature of nature. If you don't understand nature, you can't do medicine. You shouldn't do medicine. We are not in the world of engineering where we are making artificial things. What we're trying to do is reproduce nature, which is us. And then from that understanding, understand how within nature as a whole, we as a component of nature, when we don't do the right things, actually succumb to disease. And
thank you.
I'll leave it at that.
Yeah, what do you think, Greg?
I really love this comment, and I'm really happy you said this, because I'm in complete agreement. And if I could just quickly share an example from anesthesia here to support your point. So there's a drug called codeine. You probably all heard about it. It's a pain medication. And your body, in theory, converts codeine into morphine-like substances, and it gives you pain relief. And if you look at codeine on populations, codeine is a pain reliever. And it's pretty good at reducing pain. They used to use it a lot for tonsillectomies and stuff. But then as we came to better understand how the body responds to things, we learned that everybody responds to codeine differently. There are some people that they don't metabolize the codeine into the morphine-like substances, and they get no pain relief at all from it. Zero, like it does nothing for them. There are other people that handle the codeine the way they should, and they get pain relief from it. And there are some other people that over metabolize it and basically create too much of these morphine-like substances, and they get a huge amount of pain relief. And then they also get the complications that come along with this, like respiratory depression and death. So if you were to do a huge randomized double-blind placebo-controlled study of codeine on a lot of patients, you'll find that overall it's a pretty good pain reliever. But if you subdivided it, you'll find that some people are dying from it, some people are getting zero benefit at all, and other people are getting nice pain relief from it. So overall, so you're right. Like a lot of these studies, I think, fall victim to this. And then we have, as you said, which I completely agree, we have these artificial categories like African American or white or whatever. That means nothing. And so it's very difficult. And I kind of tell people that I always laugh, because I agree with you, like a lot of these studies are not worth the paper they're written on, because they're looking at populations that may not reflect your patient. And they're averaging out multiple different populations that may have nothing to do with the person in front of you. And just because it's sort of like in codeine's case, overall on average is a good pain reliever, for your patient it may kill them or it may do nothing at all. Yeah. So you're absolutely right. And this is a very important frontier of science.
Let me say three things here. One, sometimes a single case study done by a really good physician has more value than these very large studies that statistically average out everything else. As I have tried to correlate our data with the clinical world, in fact, I have found that the individual physician study that publishes a case study can be incredibly beneficial for two reasons. One, it's called construct validity. It's a human being, number one, to begin with, so it's relevant. Two, an astute physician makes observations that you can explain from a molecular basis. Number one thing. Second thing, by the way, you may not know this, you're talking about codeine. I'm in fact studying, have been funded by NIH on a 2-year fast track SBIR to take care of the respiratory distress on opioids, but in the brain and in the brain stem. The third thing I want to comment about, you brought up Viagra. You have to take a look at the lawsuits. It causes melanoma. It's not an innocuous drug. There's a class action lawsuit going after Pfizer now, showing that its repeated use actually promotes melanoma. So, the point of this is when you're looking at complex systems and you bring about a drug that is only addressing one part of the complex system, you cannot predict its effects on everything else. And that's why today we have to look at medicine through the lens of new tools that allow large data sets from within the various diseases, and also the mathematical deconvolution of those data sets to allow us to understand how a drug, predict how a drug will or won't cause all of these effects. It's doable. We are in a new era of this side of medicine that I'm promoting. You can take a look at my company, NeuroXtem, if you want. And I really appreciate everything you said. You brought about some of, through your cynicism, you pointed out all the fatal flaws of medicine, and you're a practitioner because you're seeing it in real world. The good news is addressable.
By the way, Anand, in addition to your effect of genes and environment and nutrition, the fourth thing that I would have liked to add was amount of sleep you take. That just the 8 hours of sleep versus 7 hours versus 6 hours can make a day and night difference in your health.
You're absolutely right. We know that. We know that with respect to treatment of things like diabetes. Okay,
that's in my book, by the way.
Direct connection between circadian rhythms and metabolism. So you're correct. Absolutely correct.
Sounds like you got some fans that are already reading your book.
You just promoted my book. Thank you for that.
Why Doctor Skip Breakfast? Check it out.
Why Doctor Skip Breakfast?
All right. Maybe one burning question. We need to get Greg back on the road. Soraya, did you have a question?
Yes, I have a question. So it was a great talk, Greg. So
Thank
the
you.
question I have is that we know that a lot of these experiments are not repeatable, but why is it that the scientists are not encouraged to publish all the failed experiments along with their success story? Because when we look at any published paper, we see only the success story, you know. And
Right.
if we can learn even the failures, you know, probably we will avoid, we will save time by not repeating the things, you know, the experiments.
I agree, and you're 100% right about this. And so this is a problem. So the stuff that's really interesting. So most things that are published are positive results. You know, they were looking for something and they found it. It's rare for negative results they publish unless it's about a really important thing, like you thought a drug worked and you find out it doesn't. Almost always results are positive, and this publication bias is because, frankly, the journals, they want to sell copies. I mean, this kind of goes beyond, you know, people who are smarter than I am on this could probably explain it better, but the journals want to sell more of these journals, and you'll sell more if you have interesting stuff. And there's tons and tons of stuff that's submitted to the journals. I mean, they really just get an avalanche of things. I mean, if you've ever done a study and you submit it for publication, you're lucky if you got it published, or it's really good. A lot of times you do stuff and it never gets published. And that's because there are way more people trying to get published than there are spots for publication. And so as a result, the journals have to be selective. They have to kind of pick, I'm making up this number, but maybe 1 out of every 50 or 1 out of every 100 things that's submitted to them, they could publish. And so obviously they're going to choose to publish the sort of interesting, positive results. And, you know, I kind of understand that. But the problem is it creates this false impression that everything works, and we don't see the stuff that doesn't work. And so, you know, like I said in the McDonald's example, if there's one study that shows it's healthy and 100 studies that show it's not, you'll see the one study that shows it's healthy, and so you're going to walk away with a false impression. And I don't know the solution to this. You know, I don't know how we can get more stuff out there with also not having more garbage out there. That's something that you probably need someone smarter than I am to solve, but it's definitely a real problem, and it's not an easy one to solve, frankly. But it has to do with sort of the process we use now to distribute scientific results.
I think it's going to take a community. We've got a great community here, overlapping with other communities. Keep bringing the right minds together. I think we'll get closer to the truth. Greg, I want to get you back on the road. You were awesome today. Learned so much. Really appreciate it, and appreciate the engagement from those of you on the call as well.
Yeah. Thank you, Greg.
Oh, thanks.
Exciting. At least and thought provoking.
All right, let's think about it. Let's stay connected.
Thank you, Greg.
Enjoy
Safe
your
travel.
Of course. Thanks. You're welcome.
Cheers.
Thank you.
Bye.
Safe travels.